Hey guys, I know with the coronavirus, we're all sitting at home, bored out of our skull, and I'm working from home right now. So we're actually filming this. My beautiful assistant is filming this in the infamous Almart. So I do have a little bit of a whiteboard here, and I just felt like you guys may need a little extra information on blood typing. For the most part, you're doing this, remotely, and you're looking stuff up, you're studying it. But this is what I would normally do in the lab, and hopefully this will help you understand blood typing. And then maybe I can do something with the plates that we have in the lab manual. If you notice, we have these little plates on there for blood typing, and I can explain those to you. All right, here's my 10-minute thing on blood typing. This is a red blood cell. I don't have any red pens. These are all red blood cells. And we have 4 different types of blood. We're going to put the type here. I'm going to make it another little heading here. Antigen, and this stands for antibody. AB is antibody. So in blood typing, all of these blood types have the same flagpole. They all have the same flagpole. And what this flagpole is, and what I'm going to attach to it, is called an antigen. And an antigen, running out of board already, is a chemical that can cause or will cause an antibody to be formed. Now, in the case of a normal cell has these as well. And I'm going to draw it like a flag. This is the easiest way I can explain this. It's like a flag flying. And in this case, this is my flag. So every cell in my body is marked with that flag. And again, that is an antigen, antigenic marker. In the case of your normal cells, they are called major histocompatibility complex proteins. I know that's a big long word, but we call them MHC proteins. Don't write any of that down. If you have somebody else's cell, they're going to have a different MHC protein. And those MHC proteins, if put into my body, my immune system is going to kill them. So that's the basis of this discussion. So let me see if I can erase this off this old board here. In blood typing you have the same thing, but they're sugars basically instead of proteins. So if we have, for example, blood type A, we have 4 types: AB, AB, and O. This is what is known as the ABO blood typing or ABO blood grouping. This is only one type of blood typing. There are several. For example, if you go in and have a transplant done, a kidney transplant, liver transplant, they may do all different types of antigenic typing to make sure you match. In this case, we're just trying to make sure we get the right blood, for example, if you have a surgery or something. So if it's type A, that blood type's flying the A flag. That is the antigen. So the antigen is also A. One other thing, we only have 2 antigens total in this blood typing. We have one, the other, both, or neither. So you either have A, B, both of them, A, B, or neither, O. So in the case of B, your flag is going to be B. The antigen is going to be B. In the case of AB, it's going to fly both flags, A and B. And in the case of O, it does not fly a flag. It does not have an antigen. So here we can say A and B antigen. O has no antigens. They're used to this. Erasing with my finger here. Another thing with this blood typing, A hates B and B hates A. So in A blood, it carries an antibody that is against B. We call that anti-B antibody. That's a hyphen, anti-B. B hates A, so it has anti-A antibody. AB has no antibodies. And why is that? let's go back to the A and B. If I put B in an A person, is it going to recognize its flag? No, it does not. That's why it has the anti-B antibody. It is going to fight against that unknown antigen. Remember, an antigen is a chemical that can cause an antibody to be formed. So if I put the wrong antigen in that person, it's going to fight it, and it has anti-B to do that. Conversely, if I put A blood in a B person, it has anti-A antibody, which is going to be against the A antigen and fight it. What if I put A in an AB person? Does it recognize the flag? Absolutely, it's flying it. How about if I put B in an AB person? Does it recognize the flag? Absolutely. It has no antibodies against that. What if I put O in an AB person? It's not even flying a flag, so it is not going to create an antibody response either. This is why we call AB the universal recipient. Hopefully you understand that. O, on the other hand, is not flying a flag. It just has the flag pole. It has no antigen. But if I put A in an O blood type, will it like that? No, because it does not recognize its flag. If I put B in an O, will it recognize that? No, it is not recognizing its flag. It will fight it. What if I put AB in there? It's got both of them, so it's going to hate it even worse. So in this case, we have both anti-A and anti-B antibodies. And hopefully that makes sense. So basically, O hates everybody, kind of like me. I've said that in class. You can put O into anybody. Why is that? Because it has no flag. So this is not going to fight it because it does not recognize any flag. No flag, no flag. It can go in all these. So we call it what? The universal donor. However, O can only take its own blood back. Why? Because it will not recognize any of these antigens and it will fight them. And that is basically blood typing in a nutshell. I'm going to try to, actually on Blackboard I have posted a separate file that says blood typing, and I have these simulated blood typing kits that we would have done with every blood type shown. And in that plate, let me go ahead and do this on the board. I guess you can watch this again and that will still be on there. So I can erase this. So you can watch it time and time again if you want to. And catch that again, or take a picture of it while you are. Okay, on the blood typing kits, you have 3 slots. This one's labeled A, this one's listed B, this one's listed RHO. Well, what the heck is RHO? We haven't talked about that. Well, for example, if I have an A blood type, which we just showed you with that A flag, RH is another antigen that can be present or it's not present. If it is present, we would call this A positive blood. If it is not present, it would be A negative. So RH is the positive or negative in blood types. It's either present or it's not, and it's just another antigen. So when we do the blood typing, if it clumps, let's just do it like this. This is not red, but if it clumps in the plate, and you'll see it, it'll have like little white specks. I'm having a hard time doing that with this. Let me get my other marker out here. The blue is going to stand for clumping, if you can see this. So if it has little clumps in it, which will look more like sort of like oatmeal or something, or cream of wheat, if you know what that is, that means that antigen is present. So in this particular plate, this is an A negative plate. Why? Because you have A present, you do not have B, and you do not have RH. How about if we, staying with our same analogy about A, what if we do this? I'm sorry, I can just do this on the other one up here. Be easier. All right, in this case, we have A present, no B present, RH present. That would be A positive. And then lastly, let me give you one more. What if they're all clumped? What if they all look like oatmeal? Then that is AB positive. Now, blood typing is something you either get or you don't. If you don't get it, email me and I'll try not to confuse you and maybe we can get it figured out. One more just before I stop here. I don't even know if I can post this on Blackboard, so hopefully I can. What if none of them clump? What do we have? We have O negative. Because again, O is not flying any flags. So no antigen, no antigen, no antigen, O negative. Look that up in your text. Your text is confusing about this. That's why I wanted to give you a little video on it. So email me, let me know you're out there. I think I've heard from one of you, so hopefully, or two of you. So this is going to end this and I'll try to post it later today. Thanks.